Today we're going to talk about the actual job of the DNA. So we've talked about its structure, we've talked about how it makes copies of itself. Today we're going to talk about what does it actually do? Why is it so important for our cells? So, what genes are, remember genes are codes on your DNA. We've been using letters for them in our last chapter, big B and little b. What it really is, it's a region of your DNA that makes a specific product, usually a protein or polypeptide. So it does say here polypeptide or RNA, but for purposes of our class, let's just focus on the polypeptide. So genes are codes for specifically making polypeptides, which are, you may not remember from earlier this year, proteins. And proteins make up a majority of your characteristics. It's all your enzymes, it's most of your hormones, it's your hair, your nails, your skin pigments, your hemoglobin that carries oxygen in your blood. Proteins are really, really important. So DNA is really the codes for making those proteins. But here's the thing, DNA is in the nucleus. And if you remember anything about cells, the proteins were made by these little dots, which were the ribosomes. Well, DNA can't leave the nucleus, so it has to get its message to the ribosomes so the protein can be made. And that is going to happen using a molecule called mRNA. M stands for messenger, so it's going to carry the message from the nucleus out to the ribosomes. And this is going to happen in two steps. Step one is called transcription, and this is where the RNA is going to make a copy of the DNA, technically just one little section. This is not the same as replication where the DNA copies it its entirety. It's only going to copy a little section of one side of a DNA, just one gene. Let's say you just ate some milk or drank some milk and you need to make lactase, the enzyme to digest the milk. The gene for lactase to make that particular enzyme will be activated, and that DNA will be copied by mRNA. It will take the message out to the ribosomes, where the second step, translation, will happen, and the actual protein, lactase, will be made, and you can digest your milk. So this is what it looks like in an overview. This is your DNA. The first step is going to be transcription. That's going to make your mRNA, technically in a little m here. And then translation, the second step where it's going to actually make this protein chain, which they're representing here. Notice that's going to happen in the cytoplasm. So RNA is going to be able to leave the nucleus, even though DNA can't. All right, so the sequence of nucleotides in DNA, the letters, this is what's most important. This is why DNA is so fantastic at storing information, is that those nucleic acid letters are like a language. And ultimately what's going to end up happening is those DNA letters are going to get turned into RNA, and then they're going to be read like a book. They're going to be read 3 letters at a time, and every 3 letters is going to be the code for making one amino acid, one building block of this protein chain. So they've done a lot of experiments, and the experiments basically showed us that the code is what's called a triplet code, which I sort of just mentioned. What it basically means is that DNA, when it gets turned into the mRNA, it's going to be read in these 3-letter codes, which are going to be called codons. So it's going to take 3 letters of code to make one amino acid. So checkpoint question. If you were going to make 100 amino acids, how many letters would you need? Well, if it takes 3 letters to make one amino acid, 3 times 100, you would need 300 bases of code. In other words, A, T, C, C, G, blah, 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 300 of those. And it would have to be specific ones. Understand that if you change these letters, you change the code. It's not going to read the same. Just like changing a word in a story would change the meaning of the sentence. So this is what it would look like. This is our DNA. It opens up in just one little section. RNA is going to make a copy. Now notice, they just kind of threw this at you. RNA does not have T. Remember, RNA has U. So when the DNA opens up and the mRNA comes in to make a copy, if it sees an A on the DNA, it's not going to put a T there. Remember, we were doing this before. We were starting with DNA, and we were like, okay, if we're going to make a copy of our DNA, what would attach to A? And we said, oh, a T. And T would be A, and C would be G, and G would be C. But this is different. This is going to be RNA making a copy, right? So if we start with DNA, A, T, G, C, RNA does not have T. So the match to A is going to be U. The match to T is going to be A, G to C, C to G. So these are pretty much the same, but no T. And so this is RNA. And then notice what they're showing here. 3 letters at a time is called a codon, and this is going to code for a specific amino acid that's going to build a protein. Second 3-letter code, third 3-letter code, last 3-letter code. Okay, and it turns out that this genetic code is pretty much universal. So, in other words, for example, people who are diabetic have to take insulin. Insulin is a protein that your DNA codes for making. Turns out in diabetic people, the cells that make that protein are destroyed. So what they've actually done is they've taken bacteria, they've taken the code to make insulin, they've inserted that DNA code for making insulin into the bacteria, and now you have bacteria that produce human insulin. This is genetic engineering. And now people who are diabetic, they can get insulin that they need. By the way, you might say, well, what did they do before? Oh, well, people who were diabetic had to take insulin from cows and pigs and horses. That was the insulin that we used. Their insulin is similar to ours, but it's not exactly the same, so it wasn't super efficient. So now we've basically genetically engineered bacteria that can make human insulin. And why does that work? Because if the gene codes for making insulin in us, it'll code for making insulin in any organism you stick it in. So you can stick all kinds of genes. That's how we made glowfish. We've taken the genes from a jellyfish for glowing, stuck that gene into fish, and the fish now glow because they now have a jellyfish gene inside of them. They are transgenic organisms, or sort of like GMOs, basically. So that is kind of important. So this is one version of the chart. This is actually not the version you're going to see on your test, but some people think this one's easier to read. So if we have an RNA sequence, CCA, HCG, and they want to know what these are the amino acids that build the protein. They have funky names, but that's them. So CCA is our first codon. So here's what you do. You take your first letter of the codon, you start in the middle. So we found C. That tells me it's going to be this quadrant. Our second letter is C, so now we know it's going to be this section. Our third letter is A, so it's going to be proline, or pro is going to be our first amino acid. Our second one is U. So here's U. Move out. That tells us it's this quadrant. Second letter U. Third letter U. Phenylalanine. PHE. And the last one, ACG. So A tells us it's this quadrant. C is our second letter. Third letter, threonine. THR. Here's another version. This is the version you're going to see on your test, so you should be familiar with how to read this one in particular. So what you're going to do this time, AUG, you get your first letter of the codon from here. So here's A. That tells me it's going to be this section here across. It's definitely going to come out of this. Second letter U, we go to the top for our second letter. See, second letter. Now, U has got to be this column, so now we're down to just these 4. And then our third letter is G. You get that one on the right, and that codes for MET. Second one, CCC. First letter C, this row, second letter C, this column. And check this out. You don't even need to know the third letter because they all code for the same amino acid. So our second one is pro. And that's actually something really important about this chart. There are actually 64 codons, but there's only 20 amino acids. So a lot of the codons code for the same thing, especially the third letter. Like notice here, the third letter doesn't even matter because ACU, ACC, ACA, ACG all code for threonine. GCU, GCC, GCA, GCG all code for alanine. So the code is very repetitive, and that actually helps because if we have a mutation, maybe it won't even hurt us because it won't change what it codes for. If it still codes for the same thing, it's sort of like inserting a synonym into a sentence. If the sentence still has the same meaning, maybe there won't be a problem. There are 3 stop codons. If a code got changed to a stop code, like let's say the code was supposed to be UAC, but it got changed somehow to UAA, stop is obviously not an amino acid. That's actually what tells the protein to stop being built. a lot of genetic disorders, what happens is that the DNA has a mistake, and when it goes to make a protein, like somebody say is lactose intolerant, maybe the problem is that they have the gene to make lactose, or lactase, sorry, to break down lactose, but they have a mutation in the gene that they have a stop codon in the middle. So it goes to try to make lactase, but it reaches the stop codon, which says stop here, and it never reads the rest. And so you don't make that protein. And most genetic disorders are problems with exactly that kind of thing. You make the wrong protein, or you don't make a protein at all, because of the DNA has a mistake, and if the DNA has a mistake, the mRNA is going to be wrong, right? Like this had to be coded for by a specific DNA, TAC. If this DNA gets changed, then the RNA is going to be different, and the amino acid may be different. All right, you don't have to necessarily know the name of the enzyme, but there's an enzyme called RNA polymerase, and very much like DNA polymerase builds DNA, RNA polymerase builds RNA. We're not going to talk much about the promoter and the terminator, but this is sort of like our start, and terminators are stop. So this is the process of transcription. And this is what they're showing. So the DNA opens up. The mRNA is going to make a copy of just one side of one section, not the entire DNA, not both sides, just one little part, one gene. And then at the end, the RNA is going to leave. Now, we're not really going to talk too much about this either, but the mRNA that's made, because it's going to be a messenger RNA, it's going to carry the information out of the nucleus. It actually goes through something called splicing. It's sort of like if you had an essay and there was a word limit, you had too many words. You might go through your essay and edit it, take some of the words out so you could get, try to keep the meaning but lower the number of words. It turns out you've got these non-coding segments in your DNA that become part of the RNA, and they get cut out. They're called introns because they're left inside, and the parts that leave are called exons because they're the ones that actually end up exiting the nucleus. And then the mRNA gets something called a cap and a tail. Basically, these protect it from getting broken down once it gets to the cytoplasm. Not really a big thing for your test to know about the splicing. The important thing is transcription happens in the nucleus. The mRNA is making a copy of a section, a complementary match, I should say, to a section of the DNA. This is just showing that splicing thing. So these are the introns that are getting taken out. The exons get sealed together. It's actually cut by something called spliceosomes, which sounds like I made it up, but I didn't. And it gets a cap and a tail, and it heads out to the cytoplasm. And then, when it gets to the cytoplasm, our second step is going to happen. This is called translation. What's going to happen in essence is the ribosome, which looks like this, it has two pieces, is going to hold the mRNA in place. So this is our mRNA strand, sorry. And imagine this is my first codon, AUG. Turns out there's a tRNA, it's actually shaped kind of like a T, and it has something called an anticodon. It's the match to this, which would actually be TAC, but you don't have to know that. Actually, sorry, UAC, no T's in RNA. The other side of the tRNA has an actual amino acid on it. So it's going to come, it's going to go, oh, AUG, I'm the correct tRNA for that. I'm going to drop off my specific amino acid, which happens to be methionine. And then this mRNA is going to slide down, and the next three letters are going to be read. And then the correct tRNA will come in with an anticodon match and drop off its amino acid, and these will connect. And this will leave, and then the next one will come and drop off an amino acid until it gets to a stop codon, which will basically mean it's finished. So this is what tRNA looks like. It actually looks like a letter T in a lot of ways. This is the anticodon. It's a match to the codon. The codon's the part I really want you to know about. That's the one on the mRNA. That's the three-letter code that codes for an amino acid. The tRNA has a match called the anticodon. You might just want to know the word. And then on the other end, an amino acid will be attached here, a specific one. So this tRNA may only carry methionine. So that's how it's going to know what the order is going to be in the protein. Ribosomes themselves, they're made of something called ribosomal RNA. And again, they actually have these binding sites where the tRNA and mRNA can sort of get together to build the protein. So really, ribosomes, when they say they make a protein, what they really do is hold the mRNA in place and allow the tRNA to drop off the amino acids. So this is what it looks like. mRNA, 3 letters at a time, and tRNAs are coming in and bringing the correct amino acid to each spot. You don't have to know the steps, initiation, elongation. Just know it has a start and it has an end. And I skipped a couple slides because I really wanted you to see this one. Something called the central dogma of science says that this is the way it codes. Oops, DNA to RNA to protein.